Yeah, mycorrhizal fungi, lessons from green tree retention. I'm going to touch on 3 studies I've been involved with, rather briefly for each one due to the time constraints. But before that, I will also review what mycorrhizal fungi are and why we as persons interested in forests care to know about mycorrhizal fungi. So we'll go, or are we, no, we're not going. Oh, wrong touch screen. I'll use mine. Okay. So mycorrhizae are a mutually beneficial symbiosis between the roots of most plants and fungi. And we like to emphasize the fact that it's necessary for these particular fungi to complete their life cycle, being mycocentric. Trees or other vegetation supply carbon from photosynthesis. In exchange, the fungus provides minerals, particularly phosphorus, and water from the soil. And mycorrhizae are essential for the survival and growth of most coniferous forest trees and other kinds of vegetation. I'm going to entirely ignore probably the most prevalent kind of mycorrhizae on a worldwide basis and just focus on the type of mycorrhizae that are important in our forest ecosystems in the Pacific Northwest, those being the so-called ectomycorrhiza type. And you can see from this list of plant families why the ectomycorrhizal type is important in our system, because you see here pine, oak, and birch families are what we have to deal with mostly, particularly on our west side forests. The anatomy of a pine ectomycorrhiza, this illustration diagrams the ecto part, the fungus on the outside of the root. So here the so-called mantle or Hartig net is the interface where this exchange, you might say one-for-one exchange, a certain amount of nutrients for a certain amount of carbon from the tree, nutrients from the soil, and water from the fungus. That's where it takes place. There's no actual penetration of the root cells by the fungus in this type of mycorrhiza. And here's just an illustration of the typical pine ectomycorrhiza. And the pine feeder root typically is dichotomously branched, like you see right here. And the white stuff on the outside of the feeder root, that's the fungus enclosing that little feeder root like a sock on your foot. Or if you had toed socks, so much the better. And here's just a typical Douglas fir ectomycorrhiza. You can see it has a different shape to it. It's what we call a pinnate shape. And we're going to come back to this fungus because this type of fungus on the root is really one of the most important ones for Douglas fir throughout the Douglas fir region. Now, when I say they're critically important, that comes not just as an opinion, but from actual studies. Here's Douglas fir seedlings in a nursery after being planted into soil that was sterilized, and then 3 years later you have these little islands of 3-year-old seedlings, and you can see in the middle of this island some of them have grown to be perhaps 8 inches tall after 3 years, but on the periphery of that island, and typical of most of the nursery as it turned out, they lost their entire crop. The stunted seedlings were only an inch or 2 tall after 3 years, and investigations showed that they were non-mycorrhizal, whereas the ones in the middle here, where they'd started to grow, were mycorrhizal, and then that fungus could spread root to root and start to help the trees grow. It's just a close-up illustration of the, okay, there was. Now we're going back to your movie for some reason. Hello up there. Yoo-hoo. I could hold this up so you could look at it. I don't know. Just went away.
It's not even connected.
Well, yeah. Try it again. They cut me off. Oh, okay. Thank you. We're back. Okay, here's a close-up illustration of the extent of the mycelium supporting one little seedling, here radiating out from the root system into the surrounding soil. Some of the fungi then that, well, let me say some of the sporocarps, that is the mushrooms that come from this symbiosis, are familiar to us even as special forest products, such as the chanterelles that probably everybody in this room loves to harvest, and chanterelle season is upon us, thanks to the rains. The American matsutake, or pine mushroom, and throw in this slide up here to draw your attention to a poster we have out there on a new thinning study going on to look at the effects of thinning over snow to ameliorate any negative impacts on the fungus in the soil that supports the matsutake out in the crescent area. And not to mention our most high-valued fungal product from the forests in the west side Pacific Northwest, the so-called Oregon black and white truffles, here shown at a purveyor's stand in the Oregon Truffle Festival held annually in Eugene, Oregon. Photos by Dave Pilz, and many of you remember Dave, who worked in the Northwest for many years. So I hope I've established a little bit reminder of what ectomycorrhizal fungi are and why we're interested in them in the forest. And particularly, we're interested then in the biological legacy of green tree retention on ectomycorrhizal fungi. In a context of science-based management, we've heard these terms before, adaptive management, so you want to use science and you want to be able to be flexible as science brings new knowledge to the management toolbox we've heard about. And then we also have to be thinking at ecosystem and or landscape management contexts, cumulative effects over broad scales of the region. And that's a difficult challenge when you're looking at what are essentially cryptic or hidden microorganisms. So the first study, I'm going to just emphasize one lesson we learned from in terms of green tree retention was the old LTEP program. We looked in particular at the Siskiyou Integrated Research Site, and we were comparing the late Searle and the treatment versus early Searle. Just a landscape shot of the sites across the way there. Interior shot and some little aerial photo comparing these two treatments. Some of our volunteers helping us look for the truffle fungi. And this was the one finding I wanted to emphasize from this particular study for you. This type of truffle, Archangeliella camphoraita, is rare, and we happened to find a fair amount of it in that particular Pistol River basin on our study sites. So we had the opportunity to study its response to the green tree retention that we would not have other places. And it's listed for special management under the Northwest Forest Plan. So it has some management interest in that way. So for a little bit of context, in the control here, try that again. This is a normal amount of truffle biomass production from a number of studies that we've done over in the region. 3 kilograms per hectare dry weight. And then in the late Searle treatment, and we weren't really provided with the final numbers on what that meant in terms of basal area retention or canopy cover, but I'm estimating, just looking at the aerial photos, that Perhaps it was 20% basal area retention based on what was there before. So it dropped off more or less proportionally, and that makes some sense because the carbon supply has to feed the fungus to make the truffles, or the sporocarp. However, with this particular species, Archangeliella camphoraita, when we looked at it specifically in the control versus the late cereal areas across the 3 blocks, you can see the response was variable amongst the blocks, but in all cases, the late cereal treatment kept this species of interest on the landscape. So this is why our conclusion was that green tree retention can help mitigate the effects of, or help promote the objective of maintaining the certain organisms on the landscape. That was based on one sample 3 years after the treatments. We don't know the current condition, haven't been back. Okay, moving on to the demo project. Rob Ryby showed some results from that this morning. Long-term, enhanced late successional, and again, green tree retention objectives. Here's a diagram of the layout. And from an aerial shot, you can see some of these treatments on the ground. So here's the 15% aggregated retention. There were a few wildlife trees left that were still alive in the first 3 years following treatment. There is a 40% retention right in there that you can hardly tell. That 60% basal area was removed because of the oblique angle. Here's the 40% aggregated retention, 75% aggregated retention with these 1 hectare circles cut out. And then over here, 15% dispersed retention. The scale of our investigations were at the treatment unit, and one of our response variables then was species richness. And we also were able to match up, in many cases, species richness based on the traditional approach with sporocarp production, that is the fruit bodies of the fungus, but now we can use both morphological and molecular techniques to look at the actual organism on the roots from soil samples. So we were able to look at both of those ways of assessing ectomycorrhizal fungi. And this is an example from a truffle-forming fungus. So we looked at mushrooms above ground, truffles below ground, and roots. So in terms of the roots, the ectomycorrhiza type species richness, we essentially saw 2 response patterns. So here in the bottom, a species accumulation curve, that's in the 15% dispersed retention, and the presumed clear-cut areas between the aggregates, although they weren't entirely clear-cut and burned. There were still some green trees there too, but they grouped together. And then the other treatments grouped similarly to the pre-treatment data. So we had 3 years pre-treatment data, 3 years post-treatment data. How was it that we were able to detect roots in these green tree retention areas that were heavily harvested? Well, for scale, we have a meter stick right here. You can see this long lateral root going out well away from the tree. That was a tree that blew over, but it was one of the retained trees. And so we were able to detect live roots well away from individual retained trees. And in this diagram here, we see the results of that. Underneath the retained tree canopy, the species richness was essentially the same as it was before treatment. Whereas away from the tree canopy and well into the matrix, live roots were still present, but at about half the density. Well, shouldn't say root density, but species richness dropped off significantly. And this is to illustrate here that even a stump out here might still be harboring live trees through root grafting. The roots underground are heavily grafted to each other, so an individual live tree can actually extend its influence quite a bit beyond its immediate crown. And this is also illustrated here in these results, where the inside crown data species accumulation curve matches the pretreatment data perfectly, whereas the other locations group together in another pattern. Okay, switching to sporocarp production, that's the mushrooms and the truffles. Species richness, by looking at sporocarp production, we have 3 patterns. Remember, looking at the roots themselves, there were 2 patterns, but the expression of reproduction, or the production of these sporocarps, fell into 3 patterns. So we have the control and pretreatment condition coming together over here. Then another group of 2 things, the aggregates, those are those leaf areas, and the 40% dispersed. Species accumulation curves track each other quite well. And then finally, the 15% dispersed and the harvested areas track each other at a rather low level of species richness. And remember, most of the trees have been removed from those areas, so it's not a surprising outcome. And yet, there was still a preservation of some of these organisms on the sites by green tree retention, even to the extent that they could form reproductive structures. And then I want to contrast just briefly the mushroom production versus the truffle production, because I think it gives us some important insights on some evolutionary processes in our forests. So just focusing here on the 75% aggregate retention and looking at the significant drop-off in mushroom production compared to the controls over here, even though we were only looking in the retained forest around those cutout areas. We're not even looking in areas where there weren't any trees, but looking in the aggregate around those cutouts, mushroom production fell dramatically. Now, in the 100% retention, you can see some annual variation. So this is the average over those 3 years before and 3 years after. That's year-to-year variation in mushroom production, but that's significantly different than the drop-off in the 75% retention. If we look at the truffle production, however, you can see that the pattern of difference in the year-to-year variation is not different between the controls and the 75% aggregation. That leads us to the conclusion that no one had really come to before, that these gaps in the forest that have a negative effect on mushroom production in terms of reproductive effort of these organisms did not have that same effect on the truffles. And that's why we postulated in another publication that gaps in the forest are one of the drivers of the evolutionary processes from mushrooms to truffles. These underground mushrooms actually almost all have mushroom-like ancestors. So a truffle is an underground mushroom, and it looks like microclimatic conditions in the environment are a driver of that evolutionary process. And what we do to the forest probably affect that. So the rarer species were disproportionately affected. However, the rare truffle was maintained on the sites. A couple of corollary conclusions there. Disturbance, and I didn't touch on fire because I had to keep it short, but from silvicultural practices or lack of disturbance due to fire control policies, management effects. Decision makers should be thinking about these things. And these include changes in community structure at landscape scales, which is going to be very difficult to get at or model. And this balance between the mushroom and truffle life form, how is that being affected at the landscape scale? So how much time do you have for me? My counter reset. That's about it? Or a little more? Okay, well let me touch on actually what was going to be this one. Spore inoculation of Douglas fir seedlings, bridging basic and applied. And it's been postulated that seedlings inoculated with this particular fungus, Rhizopogon vinicolor, well, it's actually been seen on harsh sites that can affect survival. And we did a little test and found that the inoculated seedlings had more roots of this inoculated type on this harsh southwest but green tree retention site. So we took upon ourselves to do this experiment, comparing these things, looking at some basic science questions, off-site inoculum versus local, a couple of on-site effects there in terms of the different logging disturbances. Here we are inoculating the seedlings. This is the site that they were planted onto. It's a green tree retention site incorporating both aggregate and dispersed retention. The dispersed retention was probably about 15%. It was considered a regeneration harvest, and there were burn piles. And I'll just click through some of this here to get at the final conclusion. 3 years then passed, and at that time there was no treatment effect in terms of our inoculating those seedlings before we planted them out onto the site. No difference. You can see the C here is the control. So there's a control treatment there, control. Those were all the uninoculated, and there's no difference across all of these in the amount of the mycorrhiza that we had tried to put onto those roots on that site. So our conclusion there was that the inoculum did not affect seedlings, mortality, growth, or inoculum levels, and that green tree retention retained a sufficient legacy of inoculum, either as living mycelium connected to those green trees or as spores in the soil to rapidly colonize the bare root Doug fir seedlings that were used in this experiment. Okay, thank you.
Time for questions. I have one that's been on my list. When you think about mycorrhizae as a network connecting trees and old trees and seedlings, as you kind of hinted, if you think about marking thinnings, are there some trees that are better connected than others? Is there something we can look for to facilitate that, or are all trees created equal?
All trees are not created equal. There's a really nice study out of British Columbia that addressed that, and if I had time I could pull that up on here, but we don't. I can just say envision a network model in which the size of the tree is represented by the number of connections it has to other trees via the mycorrhizae, and it's a heterogeneous, spatially explicit model. And definitely there's some trees that have a lot more connections than other trees. But the second part of your question was, can we predict which trees those are? No, not without doing a lot of work. Yeah. Just send money. A lot of that around, right? Okay.